Okay. DNA methylation age of human tissues and cell types.
I'm Kevin.
I'm Isabel.
I'm Coven.
I'm Sienna.
All right, so what is DNA methylation? I think before we get into this, we should address what epigenetic changes are or what epigenetics is in general. So epigenetics is basically the link between genotype and phenotype moderated by the environment. So epigenetic changes include histone acetylation, microRNA interference, and of course DNA methylation. So these changes are changes that occur like they're not inherited from your parents, like DNA is. These are changes that are influenced by your environment that can indirectly affect your genome and gene expression and consequently phenotype.
Mhm.
So what is DNA methylation? In our genome, we have four nucleotides, right? A, T, C, G. DNA methylation is on cytosine nucleotides. They get methylated into 5-methylcytosine. And the significance of this is that when the cytosines are methylated, it kind of makes, like, let's say this is your DNA. It kind of makes the DNA coil in on itself tighter, which makes it harder for enzymes such as DNA polymerase or like RNA polymerase to replicate or transcribe. So it affects the activity of that segment of DNA and then therefore gene activity.
Mhm.
So how they actually measure these methylation changes are through, I think, modern method. The modern method is whole genome bisulfite sequencing. So what they do is this cool thing where they use bisulfite. They treat it with bisulfite, and it's like a whole chemical process. But essentially, unmethylated cytosines are converted into uracil, and then methylated cytosines are converted into thymine, and then they can take the ratio of uracil to thymine to determine like what percentage of the DNA in that region is methylated. So how DNA methylation is actually studied, methylated regions are clustered into CPG sites, which are sites like in the genome, which contain like hundreds, tens, hundreds, thousands of CPG repeats, which are cytosine nucleotides followed by guanine. And these CPG sites or CPG islands are typically located in promoter regions of genes or introns or exons or like transcriptional start sites, like near those regions.
Mhm.
So when these sites become percentagely modified or methylated, they can affect the activity of that gene.
Okay, so what is an epigenetic clock? Essentially, it's like measuring your age based on like the changes in methylation. So like Kevin said earlier, it's determined by well, it's modified a lot by your environment. So this includes smoking, drinking, living in a polluted area may affect your lungs, kind of thing. Or protective factors would be taking care of your body, and that would help slow down the aging process.
Yeah. So in the big picture, your methylation patterns at these CPG sites change as you age. And then using these patterns of changing, you can build a model to predict your age, if you will, from
Yeah.
these methylation changes.
So like
Your
for
body's
example,
age. Yeah.
yeah. So like for example, let's say at a certain significant CPG site, at birth you might start out with like 0% methylation, right? And then let's say when you're like 100 years old, you're at 100% methylation, right? So over time, like obviously it gets increasingly methylated. Let's just say for this significant CPG site, right? So you can build a epigenetic clock, like this model based on that. So eventually, if you put in like this methylation amount at this CPG site, like let's say you're 60% methylated, right? That clock will then tell you, oh, I think that you're 60 years old, right?
Right. Yeah.
It's a good predictor of things like death.
Yeah. This ties into machine learning, which we'll get into more. Okay, so going into the actual study, this was like a study. They didn't generate their own data, they just got data from public sources, but they got it from 82 different studies. They went
Like
online, searched up
crazy.
82 papers, and downloaded the data from that, cleaned it,
Yeah,
like pre-processed it, and then used it. I like the very first video of our series, like we talked about how feasible it is for high school students to actually get data from online sources and like, download it, do their own type of thing, try to extrapolate new information from it. But things of this scale, like 82 data sets, that's actually insane.
Yeah,
They
I'm
this
probably
pretty
guy-
found
sure.
more, and they only had 82
Yeah,
usable
like
data sets.
Steve Horvath, the guy who did this, like I'm pretty sure this paper only had his name on it. So like,
Yeah.
he is-
Also, like, I'm pretty sure
Literally.
Kevin, you like emailed Horvath and was like, "Oh, hey, like, how can I get access to this?" And it was like super complicated. Like,
yeah, no, it's like, oh, you have to go like contact the editor of the paper to get
yeah,
permission.
like the agency, see if they release
Yeah,
like
it's crazy.
it was crazy.
But okay, so how they actually measure the methylation, like how the data set was structured, they used Illumina 27K or 450K platforms to measure methylation at the respective number of CpG sites. So the 27K platform measured methylation values at 27,000 sites, and the 450K 450,000 sites. And the Illumina 450K is what's widely accepted to be used right now. I think there's a new one. It's like 850K, and it includes like CpG sites and like, I don't know, like other stuff, but that's more high tech.
Yeah, and
But
then
out of the 82 used, he used 39 for training. So training and testing, this kind of ties into machine learning models, and 40 to 71 data sets, 40 to 71 were used for testing. And if you guys aren't familiar with like how this actually works, when you're building a sort of model that tries to predict the patterns from data to create like an age predictor, you want to make sure your training data is valid. Like you don't want to overfit your model to the training data, because maybe that training data has some discrepancies that make it like special only to that data. So you have testing data to make sure that your model works on data that it wasn't trained on. So it's new data that, and then if it works, well, you're good, right?
Mhm.
Okay, so when this guy actually built the model, he applied it to basically every value in the data set, so all tissues, and he got a pretty accurate model. He what is this, error of only 2.9 years? Like imagine predicting your biological age to an error of 2.9 years.
like
That's pretty
impressive.
crazy. And a correlation coefficient of 0.97, p-value of incredible significance.
That's crazy.
Yeah, it was a very, very good model.
Yeah, and you can see like everything fits pretty much this regression line. But I think this is where we should introduce the concept of like the age predicted by this model versus your actual age. So like, what's the difference? Is it just, if your predicted age is different, is it just wrong? Well, the predicted age is actually a thing. It's called biological age or methylation age, and it has applications past just your chronological age. I don't know, Isabel, you want to go into that?
Okay, so basically, age acceleration, like it can reveal a lot of things about your health. So for example, let's say you are a person who lives in the city, like Beijing, there's a lot of smog, and you're smoking all the time, and you generally live like an unhealthy lifestyle, right? So what happens is very likely you put your methylation data into these clocks, and it'll say that you're like 49 years old when you're actually just like 45, right? So that means that this clock thinks that like your genome is older than you actually are, right? So that can be a marker of like, oh, this might be a sign that like, oh, I'm unhealthy, like I need to make some lifestyle changes. Like additionally, and this is something that we'll get into later, like this can also reveal a lot of things about diseases. So like we all know that cancer has some pretty crazy growth patterns, right? So if you get a tissue sample, methylation, like the predicted biological age is crazy off. Like that might be a sign that, oh, hey, something's wrong here and I need to get a checkup, right? So it has a lot of different like useful applications.
Yeah, and I don't know, like Sienna or Kevin, have you guys ever like smoked or like
No.
No,
You ever
like,
get lit?
like get crazy on alcohol or
no, but
okay,
like,
but let's assume you have. You would probably have a higher predicted
Methylation. Yeah.
biological age than your actual age, which means that you'll die sooner. So watch out.
I know if you guys have seen those like drug PSAs that maybe they should in like elementary or middle school, they always talk about like, oh, if you like smoke or drink something, you age faster.
Yeah, yeah. And then
Yeah.
this is the actual biological
Yeah, yeah.
validation for that.
Yeah, like you literally do age faster. Like
Yeah.
But it's interesting
Literally.
because, oh, Sorry, I was
Oh,
gonna say it's
yeah.
interesting because I mean, our like medical technology is incredible, right? We can see a lot of these like diseases developing, and we can be like, oh, your lifestyle is not good. You eat horribly, you don't exercise, you don't sleep well, and you smoke. And it's like, yeah, you have that conversation with your doctor, but I think a lot of people don't realize the significance of this. But maybe this is what they need to scare them
Yeah.
into
Yeah,
the lifestyle.
it's not just like, oh yeah, I'll deal with it. I'm okay with being like
Oh, I get
overly
cancer, it's
obese.
okay, I go to the
But it's
hospital.
like,
No, it's like you're gonna die.
yeah, exactly, right?
Yeah, like that's one of the things in the doctor is like, oh, like your blood pressure's high. It's like a faraway thought, like, oh, that doesn't really like I feel fine, like I'm living my day
It
to
doesn't
day.
affect me now.
Yeah, but no,
Exactly.
you can see in these like clocks, or potentially you will see in the future in these clocks, like, no, it impacts you right now. Like you lost like 10 years off your lifespan. That's
Yeah.
crazy.
It's like went out for the weekend, lost 5 years. Yeah.
Yeah.
Okay,
But
so the
Um,
model before was based on all tissues, but when this model is applied to each individual tissues, it reveals some discrepancies. So the two most significant were in breast and heart tissue, where you can see like the actual ages and predicted ages don't really line up well. And a
mhm.
few explanations they gave for this in the paper was, okay, breast cancer has such a high prevalence, as we'll get into later, and also hormonal changes,
Oh
like
yeah,
the
yeah.
influence of hormones on the breast. I think all of these factors together just kind of mess with the methylation, like phenotype of the breast tissue. And also heart tissue, our heart is always recruiting new stem cells to like,
Yeah,
you know,
because you have to
do
think like your
do heart
heart's
things.
always
it,
exerting, always pumping,
Yeah.
right? You need
And then if
more
you think about it,
cells.
stem cells are new cells, right? Like they can differentiate into any type of
Oh
cell.
yeah, yeah.
You know,
Right.
like in this
babies.
case, stem cells will turn into cardiomyocytes. But these stem cells, because they're new, they would probably have like a default
Oh yeah,
methylation,
yeah.
you know, so it would mess with the readings a little bit.
Mhm.
But still, they're like pretty accurate. Like they're not that
Yeah,
far off.
like it's
They're not that off. But
Yeah, which is
maybe not
impressive.
the heart one, but it's like if you look at the breast.
But 13
yeah.
years versus what, 2.9?
But like
Yeah.
still, even if it's off in the grand scheme of things, we still have like a general trend
Oh yeah, yeah.
Hmm.
Mhm.
upwards, or which reveals the
Which is
power of
interesting.
this
Yeah.
model. Okay, so a few
It's
other observations they had in this paper. I think we don't need to go over the cell passaging thing. It's basically cell passaging is where you culture cells, and then every time you passage them, it's like a new generation.
But
And essentially, they just validated that as you passage cells, DNA methylation changes. So, analogous to growth.
yeah, because as generations go on, obviously they have more time, more methylation. So like it's changing. So it's
Yeah.
just validating that this DNA methylation as like a predictor.
Yeah. And then heritability of DNA methylation, like the traits that you inherit from your parents, it's from the DNA, but it's from like your
that's like
default
patterns.
methylation patterns as
Yeah.
well. You're born with some amount of methylation in some CBG sites.
Mhm. Which is
And
interesting because previously they thought that epigenetics, like it was just your lifetime thing, like it doesn't impact your children, but it does. Like your your patterns of DNA methylation, like they do pass on.
But
right.
again, if you have a parent who lives to like 114, doesn't mean you are going to, because your environment
Right.
plays a
Yeah.
Yeah.
massive role. So you could reverse all of this and you could
Yeah.
like screw yourself over and
Yeah,
then die
like
at
if you
40.
go into like heavy smokers or drinkers, it's not like you're gonna die. It's like as fast as
Yeah.
Yeah. Like
Mhm.
you always
well.
have the power to like change
And
that.
the genetics, it's important to know, I think the genetics could reduce like the effect of some of these like smoking
Right.
and drinking
Yeah.
and things,
Yeah.
because there are people who reach 80 or 90 and did not treat their bodies right, but without that they could have hit
Yeah.
100 easily
Just
because
like
they
don't
had
take it for
prevention.
granted, you know, just keep taking care of your body. This kind of reminds me of like, you know, rich kids, they just like, oh, like daddy's money. They just take it for granted. They don't work hard. It's
Yeah.
kind of the same
Mhm.
concept. If
Yeah,
like
like
somehow you know you have good genetics, like don't
They're like,
don't
oh,
take
I'll be
it for
We
fine.
were talking about this
yeah,
other day
like
in
it's
school. It's
okay.
like you have rich parents, their kids don't work as hard. If you have parents who aren't as rich, the kids work harder. It's
Right, yeah. It's
yeah.
the same.
Mhm.
And then also significant CBG sites here. So I think we've talked about this before. Like there are 450,000 like CBG sites mainly studied in our genome, but we can't build a model out of all those because I think we said some sites are just random.
Yeah, like you look at
Um,
some sites, there's no correlation of age and methylation. Like those
yeah,
aren't going to produce good clocks, right?
yeah. But this model selected for 353 out of these 450,000 sites that have like, oh, this site
Like
goes like this
Oh,
site goes like this site goes like
like
this
high
site goes like,
correlation.
yeah, there's certain patterns that they follow, and then they can use these patterns, input it into a model, and use it as an age predictor. Okay, so okay, this is gonna go back to the concept of age acceleration that Isabel mentioned. But first, on the left, this is a heat map of every single CBG used. Okay, well, this is the clock CBG, so this is actually 353 rows. But you can see, I think blue indicates low methylation here, and red indicates high methylation. There's not that much significance in that, oh, there's more blue than red. Like, that doesn't really matter. But you can just see some of the patterns that are exhibited here. Like these sites are red at first, and then they might turn blue. Or
hmm.
like this, like some of them, I don't know. Let me try to find one that increases. Okay, anyways, you get the idea. Like some of the sites, they start off blue and then they turn red and so on. And then these are the sites that we want to integrate into our model. And then on the right side, it shows age versus methylation age. And the slope is actually age acceleration. So the higher the slope, the higher the age acceleration. So you can see that from 0 to 20, the age acceleration is really high, but it kind of levels out as you get older. I don't know, do you guys have any explanation for that?
Like, I mean, when you're from 0 to 20, you're constantly developing, right? You're like growing at quite a like exponential rate. So I guess that's what like
Right, yeah.
Puberty, yeah.
grow older and older, kind of like you don't grow that much, right? You grow slower and slower.
It's more of
Yeah,
like when you're younger, it's like you're growing, right? So it what your environment is like changing upon you isn't as significant, you could say. Because like if you
Yeah.
could be a kid and you get like no sleep, but you'll probably still grow like enough. But when you're older, like your environment has a way more significant effect on your methylation, and that's part of the
okay, cancer. So
Yippee!
yeah, okay, so I think Isabel talked about it before, but cancer, like we know cancer is like the most dangerous disease because it has to do with our cells. Like our
Mhm.
cells don't function well.
Yeah.
Their gene expression patterns are way off. And consequently, their methylation patterns are going to be off, right? Because they control the gene expression. So you can really see that age acceleration of these cancers is just all over the place. Some age slower, some age faster.
But
And like in this one, skin cancer, number of mutations, log 10. So this will be 10, this will be like 100, this will be 1,000 mutations. Like no matter the number of mutations, the age acceleration is still all over the place. Like you can't find any correlation within this, which
mhm.
really revealed like all the intricacies within cancer. Like each cancer is unique. Each cancer has these like crazy effects on your genome that we can't really predict or working towards predicting right now.
The DNA
It's also
methylation.
a wait, go back one more slide. It's also a good marker. You see some of the ones, it has like age acceleration of 100. I don't know if we mentioned this earlier, but I think in the Horvath paper, which is the one we're doing right now, they measured age acceleration by the predictor minus actual age. So obviously, if your predictor is saying that you're 100 years older than you actually are, like that's a sign that something's not right, right?
Right. And cancer is characterized by abnormal growth, of course. So I think if you're 20 years old and your skin cells are 120 years old, that's
Oh,
pretty abnormal growth.
Yeah. Like might want to get that checked out.
Yeah. Yeah, so basically the paper is a pioneer study sort of. There was another paper before this that constructed like a real epigenetic clock, but this is one of the first in the field. So basically says that DNA methylation is a promising biomarker to study, and it has like a host of applications across all all disciplines.
hmm.
And the error rate was very low, which means it's reliable.
Oh, right. Like the overall model, I think like for humans, the variability, like it was plus or minus what, three years?
3.6, yeah,
Yeah,
3.6 years.
which
which
is
is
crazy.
impressive.
That's
Like if
crazy.
you pointed a random guy on the street, like I don't think I could give you accuracy within like plus minus 3.6.
Anyway,
Right.
if you notice that you are aging quickly and you don't want that to happen, you can address it with some currently
Come to a
arising.
clinic today and get your biological age measured.
Mhm.
Something like NMN actually reverse like your
Oh,
like
NMN was what I was
Oh
Okay,
gonna say.
yeah,
yeah.
okay.
Make sure to check out
We did a video.
JC3.
One of the
Yeah.
corners.
I think the last paper we talked about NMN, like having it as an anti-aging mechanism and like mediating oxidative stress and these metabolic pathways, it probably has some epigenetic effect as well that
Mhm.
people are not
Well,
investigating.
if it reverses your aging, I'd hope so.
Yeah,
Well, yeah,
probably does.
right.
You didn't
Well,
cover it in the paper.
also, something interesting that we discussed in the NMN paper is like, oh, like you look at growth pyramids and like predictions for the world, like our population's getting older and older. Like all these diseases like cancer and chronic diseases like are just going to become more and more prevalent. And I think this is something that can be very promising for revealing and like better diagnosing, you know.
Yeah, and just studies investigating the mechanisms of aging and like, oh, how to stop it possibly are just gaining significance in today's world. But also epigenetic clocks, they have an application where you can, okay, let's say you run an experiment, right? Like you have two normal people. One of them is your control. So you have like a 20-year span where the normal person stays normal, but then you have the other person which develops some like colorectal cancer, let's say.
Hmm.
So you can compare, okay, like this is like X equals 0, and then like 20 years later, right? And then this is the normal methylation, and then this is colorectal cancer methylation. You can compare the changes um in the methylation patterns and see like, oh, the colorectal cancer one has methylation increases in CPG sites that aren't increasing in the normal patient. So we can say that we can pinpoint this cancer affects these CPG sites,
Okay, okay.
induces these epigenetic changes, and then we can target those directly.
Yeah.
Like,
And
This
it's like
decreases the overall like variability of cancer that we're kind of scared of right now.
And because earlier we showed how even in the like general model, which included all tissue types, like there were still multiple like tissues that had discrepancies. So like it could be like building separate clocks for each of the individual tissues, or like even like individual diseases can be more like specialized ways to like diagnose and study these.
And it wouldn't be too
Yeah.
complicated to do.
No.
Yeah. And then
Just
maybe
a little time
as the
consuming,
technology progresses, maybe it'll be normalized,
automated.
just like yearly checkup, check your methylation age, like
Exactly.
see
Yeah.
like,
How are
oh,
you doing?
are you really living the lifestyle you say you are?
Yeah, like
You know,
like
like
let's go. I'm
some sort of like party
16,
monster.
but they think that I'm 12. Like,
Whew,
ooh.
I don't think that's good.
Well, if you're into that,
Okay, thank you for
okay.
watching.
Yeah.
Check out the
Yes.
NMN video. It is relevant and also very interesting.
Yes.
uh,
Please take a ponder DNA methylation and how this epigenetic clock could be useful.